Hello everyone, welcome back to Food Catalyst. We are in part 5 of the discussion on the Kerala Food Safety Officer Examinations question papers. You can download the PDF of this video through our Telegram channel, and you can also connect with us through the social media platforms which are displayed on the screen. Simplifying safety, igniting knowledge is our motto, and given are the QR codes for our WhatsApp, Telegram and YouTube channel. Without getting delayed, let's get into the questions. First question is antibiotic streptomycin is what? So the options given are protein, steroid, glycoside, and none of these. So basically the options given are macronutrients. So protein and steroid is a lipid. Glycoside is any compound that contains a carbohydrate molecule that is convertible by a hydrolytic cleavage into a sugar and non-sugar component. So it is basically a carbohydrate. So the answer is glycoside. Streptomycin is an aminoglycoside antibiotic which is originally isolated from the bacteria Streptomyces cruzius. It is the first discovered aminoglycoside. Its use is primarily treating aerobic gram-negative bacterial infections such as brucellosis, tularemia, plague, which is caused by Yersinia pestis, tuberculosis, which is usually treated in combination with the isoniazid, pyrazinamide, and rifampin. And also certain cases of endocarditis when combined with beta-lactam antibiotics. The use of streptomycin becomes particularly applicable in the emerging discussion of how to treat an infection caused by the drug-resistant Mycobacterium tuberculosis, which is usually the treatment is given by the standard combination therapy of rifampin, isoniazid, pyrazinamide, and ethambutol, which is not effective. As with all aminoglycosides, streptomycin is bactericidal, and aminoglycosides are hydrophilic, which means they are not able to penetrate the hydrophobic bacterial cell membrane. Next question is fat in the liquid state is options given are oil, sterols, wax, bacon. So the answer is oil. So the fats and oils are called triglycerides, also known as triacyl glycerols, because they are esters composed of three fatty acids units joined by glycerol, which is a trihydroxy alcohol. So this is the chemical representation. This when three fatty acids are combined with glycerol in the presence of catalyst, triglyceride is formed with three parts of water. So a triglyceride is called a fat if it is solid at 25 degrees Celsius, and it is called an oil if it is liquid at the same temperature. These differences in melting points reflects the differences in the degree of unsaturation and the number of carbon atoms in the constituent fatty acids. The triglycerides obtained from animal sources are usually solid, while those of plant origin are generally oils. I'll tell you why. The saturated fatty acids have hydrocarbon chains connected by single bonds only, while unsaturated fatty acids have one or more double bonds. Unsaturated fatty acids, which are liquid at room temperature, are considered beneficial fats because they improve blood cholesterol levels and they ease inflammation and they stabilize the heart rhythms and play a number of other beneficial roles. These unsaturated fatty acids are predominantly found in foods which are from plant sources such as vegetable oils, nuts, and seeds. There are two types of good unsaturated fatty acids. One is monounsaturated fatty acids, and next one is polyunsaturated fatty acids. Monounsaturated fatty acids are commonly found in olive, peanut, and canola oils, avocados, almonds, hazelnuts, pecans, and seeds such as pumpkin and sesame seeds. While polyunsaturated fatty acids are found in sunflower, corn, soybean, and flaxseed oils, and also in walnut, fish, and canola oils. Though in canola oils, though they are the monounsaturated fatty acids are high, it is also a good source of polyunsaturated fats. Next is omega-3 fats are also an important type of polyunsaturated fat, but the body can't make these fats, so they must be consumed. They should come from the foods consumed. An excellent source of omega-3 fatty acid is fish and even flax seeds, walnuts, canola, and or soybean oil. Next is saturated fat, which is found mainly in animal foods, but a few plant foods are also high in saturated fats, such as coconut, palm oil, and palm kernel oil. Next question is steroid nucleus contains dash number of carbon atoms. The options given are 17, 21, 18, and 16. The correct answer is 17. Here, the steroid skeleton structure is given. You can clearly see there are 17 carbon atoms. So the steroid skeleton structure has three hexagonal rings and one pentagonal ring. So it consists of four fused rings. So the steroids are the most important non-saponifiable lipids, and these compounds includes bile salts, cholesterols, and other certain hormones such as cortisol and other sex hormones, which includes both male and female. And I have given two examples. One is cholesterol and estradiol, and the structure of cholesterol and estradiol is given as well. So the estradiol stimulates the female sex characteristics and regulates changes during the menstrual cycle. Steroids occur in plants, animals, yeast, and molds, but are not in bacteria. They may exist in free form or combined with fatty acids or carbohydrates. All steroids have a characteristic structural component consist of these four fused rings. The next question is omega-3 pentanoic acid present in fish oil is options given are arachidonic acid, cervonic acid, oleic acid, and timnodonic acid. This question may make you all puzzled because there is no eicosapentaenoic acid or docosahexaenoic acid or alpha linoic acid in these options. So I'll tell you. So the omega-3 fatty acids are basically found in fish and flaxseed and in dietary supplements such as fish oil. So the three main omega-3 fatty acids are these, like I mentioned, the alpha-linolenic acid, eicosapentaenoic acid, and docosahexaenoic acid. So the other vital name for eicosapentaenoic acid is timnodonic acid. So that is what given in the option. So the right answer is timnodonic acid. So eicosapentaenoic acid is an important polyunsaturated fatty acids which is found in fish oils, and it is also the precursor of prostaglandin 3 and thromboxane 3 families. So it is basically a carboxylic acid with a 20 carbon chain and 5 cis double bonds, and the first double bond is located at the third carbon from the omega end. This OH double bond O is the omega end, and you can see this cis double bond starts from the third carbon atom. It is very important for you to know this structural composition so that you can uh, answer questions that comes out of these topics in various forms. Like the sometimes the questions can be tricky, so it is better you understand the topic completely. Next is dash vitamin is a naturally occurring lipid soluble antioxidant. The options given are B1, E, C, and B12. So the vitamin B and C are water soluble. So the option is E. E is the lipid soluble antioxidant. So this is the classification of vitamins. They are classified into fat soluble vitamins and water soluble vitamins. Fat soluble vitamins are vitamin A, D, E, and K. So vitamin E is an antioxidant that protects cells from damage. So each vitamin has many functions. So I have simplified and I have given the most important functions of these vitamins. So next is water soluble vitamins, which is again classified into non-B complex and B complex. Non-B complex is vitamin C, and B complex is further classified into energy releasing and hematopoietic. Hematopoietic has vitamin B9 and vitamin B12, which is folic acid and cyanocobalamin respectively, and the functions are given. And energy releasing B complex vitamins are thiamine, riboflavin, niacin, pyridoxine, biotin, and pantothenic acid. So, vitamin E is a plant-derived lipid-soluble substance whose molecular structure is comprised of a chromanol ring with a side chain located at the C2 position. Vitamin E refers to a group of 8 different compounds such as alpha, beta, gamma, and delta tocopherols, and correspondingly 4 trianols. So this alpha, beta, gamma, and delta forms differ with respect to the number and the position of methyl groups on the chromanol ring. The alpha forms of tocopherol and trianol have 3 methyl groups at the C5, C7, and C8 positions of the chromanol ring, while the beta and gamma forms have 2 and delta form have 1 methyl group. This explanation is very much understandable in the given structure. So in the tocopherol structure given above has this R1 and R2, and tocotrienol has also R1 and R2 groups. So if you come below here, this R1 can be substituted in alpha tocopherol. This R1 and R2 can be substituted with this methyl group, so it becomes alpha tocopherol. And if you substitute with this methyl and H hydrogen, it becomes beta tocopherol. And if you substitute this hydrogen in R1 and methyl in R2, it becomes gamma tocopherol. And if you substitute hydrogen in the R1 and R2 groups of tocopherol, it becomes delta tocopherol. Same way, if you substitute this R1 and R2 groups in the tocotrienol, it will become their respective tocotrienols. So it is better if you write down this individual structures so that it'll be easy for you to understand. So some of the functions of vitamin E. So the most important function is it is an antioxidant. It is an antioxidant by scavenging free radicals, especially peroxyl radicals and singlet oxygen, and the membrane stabilization by forming complexes with destabilizing molecules so as to prevent disturbance of the amphipathic balance within the structure. It also the physiological regulator. It is also a physiological regulator of enzyme activity, cellular signaling, cell proliferation, and gene expression, which is not directly related to antioxidant action, and also inhibition of platelet coagulation. It also prevents the diseases including neurological disorder, cardiovascular diseases, age-related eye and skin damage, and also infertility. It is a biocompatible modifier of biomaterials and medical devices. For example, in high molecular weight polyethylene used in hip and knee implants. Next question is, individual fatty acids can be separated by, the options given are HPLC, MSMS, GLC, and MR, NMR. So you should know what is HPLC. It is a high performance liquid chromatography. Next is mass spectrometry. Then is gas liquid chromatography. Next one is nuclear magnetic resonance. So the option is gas liquid chromatography. According to FSSAI, the individual fatty acid can be separated and determined by gas liquid chromatography. So, how this gas liquid chromatography works is a separation methodology. It basically employs a gaseous mobile phase, typically an inert gas like helium or a non-reactive gas like nitrogen, and a liquid stationary phase. The basic principle is It is based on the difference in the partition coefficients exhibited by the volatilized compounds between the gaseous and liquid phases. This phenomenon is observed when the desired component, which is carried through the column by a carrier gas. The testing of the purity of a substance is an example of a typical application of gas liquid chromatography, as is the preparation of novel compounds from mixtures, which is also known as preparative chromatography, and in the identification and separation of various other components, which is composed of mixtures. So the fatty acids are group of lipids composed, most commonly analyzed by gas liquid chromatography. This method is applicable to biological samples containing compounds with a chain length ranging from C14 to C24. Next question is, high intake of polished rice or other refined food items leads to deficiency of which vitamin? So the options given are vitamin B6, E, D and B1. So the right answer is vitamin B1. So here I have given the same classification again for you to understand easily. So this B1 comes under water soluble vitamins, which is a B complex energy releasing vitamin. The other name of B1 is thiamine. So the most important function is it assists in the energy production from carbohydrates. So, so basically, on harvesting the rice crop, the processing takes place in a way that the grain from the paddy crop is dehusked, which is the husk is removed. Uh, as a result, we get the brown rice. So the outermost husk, which is the outermost covering, which is called as husk, is removed, and the process is called dehusking, and we get the brown rice. So brown rice the is basically rich in vitamin. So what happened is this brown rice is then polished. By polishing, we remove the germ and the bran layer, which is basically rich in B1, to obtain a white rice. So during this polishing, the rice loses, as we lose the bran layer and germ, we lose the major important nutrients. And the one of the major important nutrient is vitamin B1. That's how this polished rice becomes deficient in vitamin B1, and a deficiency of vitamin B1 in our body causes a disease called beriberi. Next question is, dash can be measured as the ratio of carbon dioxide produced to oxygen consumed. So the options given are RMR, BMR, REE, and RQ. So RMR is resting metabolic rate, and BMR is basal metabolic rate, and REE is resting energy expenditure. So let's decipher these codes. So starting from basal metabolic rate, it is basically the amount of energy used when you are lying still but you are awake. This energy is the minimum energy that is used, that is necessary to keep your organs working. This basal metabolic rate is basically calculated for at least 24 hours at a stretch. And next is a resting metabolic rate. This is also similar to basal metabolic rate, but the energy used, this calculates the energy used to do some low effort task, such as eating, just taking a small walk without any major exercises. Okay. Next is resting energy expenditure. This is the measurement of how much oxygen a person consumes from air. Okay. Next is respiratory quotient. So here, respiratory quotient can be measured as the ratio of carbon dioxide produced to oxygen consumed. Respiratory quotient is also known as respiratory ratio. It is given by RQ. So it is basically the volume of carbon dioxide released over the volume of oxygen absorbed during respiration. It is a dimensionless number. It is used to calculate the basal metabolic rate when estimated from the carbon dioxide production to oxygen absorption. This is the formula, that is the volume of carbon dioxide released divided by volume of oxygen absorbed. It is basically calculated for a particular substrate, that is either carbohydrates or organic acid or fat or protein. So I've given for the macronutrients, the value of a respiratory quotient for the macronutrients is given. And carbohydrates are oxidized through aerobic respiration, resulting in an equal ratio of this carbon dioxide release and oxygen consumption. That is why the value of RQ is 1. Subsequently, the RQ for fat, protein, and anaerobe is zero point 0.7, zero point 0.8, and 0 respectively. If a mixture of these substrates are consumed, then the value will be collectively zero point 0.8. Next question is sum of all internal chemical activities that maintain the body at rest is, the options given are catabolism, inertia, energy expenditure, and basal metabolism. So the answer is basal metabolism, which is represented by HEE. It is a result of chemical change that occurs in the cells of a body in fasting and resting state, using just enough energy to maintain vital cellular activity, respiration, and circulation. In other words, then a minimal amount of energy necessary to maintain respiration, circulation, and other important body functions. For the measurement of basal metabolism, which is basal metabolic rate, the body must be in thermoneutral environment, a post-absorptive state, resting but conscious, quies- quiescent, and without stress. So this is a detailed explanation of basal metabolic metabolism, also known as basal metabolic rate. So the next question is, heterocyclic isoalloxazine ring is present in which of these compounds? Riboflavin, pyrimidine, purine, or cholesterol? Cholesterol, we have already seen the structure, and the rest options are pyrimidine and purine and riboflavin. So the answer is riboflavin. Flavins are basically a class of heterocyclic organic compounds. It as I'll tell you what is a heterocyclic molecule. It contains at least one ring structure composed of carbon atoms along with atoms other than carbon, such as nitrogen, oxygen, or sulfur. And also it contains these flavins contains a specific ring structure called heterocyclic isoalloxazine ring. Therefore, if a molecule contains isoalloxazine ring, it is classified as flavin. It is the structural foundation of isoalloxazine is the structural foundation of flavins. Flavins are primarily known as vitamins and coenzymes due to their essential roles in various biochemical processes within the body. However, under specific conditions, particularly when exposed to UV light, flavins exhibit pigment-like behavior. This fluorescence property of flavins, including riboflavin is vitamin B2, and its derivatives such as flavin mononucleotide and flavin adenine dinucleotide allows them to emit a yellowish pale green color when illuminated with UV light. This fluorescence is due to the specific molecular structure of flavins, particularly the isoalloxazine ring, which is responsible for their pigment-like behavior. Flavins are synthesized only by bacteria, yeast, and green plants, and for this reason, animals are dependent on the plant sources for them. Here is a list of some of the most important common flavin vitamins. So first is riboflavin, which is found in milk, eggs, meat, leafy vegetables, and important for growth, skin health, and energy production. Next is riboflavin 5-phosphate, also known as flavin monophosphate or shortly FMN. This is a coenzyme derived from riboflavin involved in various cellular processes, particularly energy metabolism. Next is flavin adenine dinucleotide FAD. This is another coenzyme derived from riboflavin, which plays a key role in energy production and various biochemical reactions in cell. So the only flavin vitamin in the list would be riboflavin. The others are coenzymes. They are some of the main, these are some of the main flavins. They play a crucial role in various biological processes. So let's break down this structure, this flavin structure. It consists of pyrimidine ring, benzene ring, and alloxygen. Okay. So look at this picture. So it has a six-membered ring structure containing of four carbon atoms and two nitrogen atoms. These nitrogen atoms are at positions 1 and 3 of the ring. This pyrimidine ring is fused with benzene ring, and it is a six-membered ring composed of entirely of carbon atoms with alternating single and double bonds. So this alloxygen and isoalloxygen, the structures depends on the specific arrangement of the atoms in the ring system. Based on this arrangement, it can be referred as alloxygen or isoalloxygen. These terms are used interchangeably and denote the same general structure basically. So it is a tri-cyclic structure as it has three interconnected rings of atoms. Okay. So in flavin such as riboflavin, the isoalloxygen ring system is the center of the molecular function. It is responsible for the molecule's ability to absorb and transfer light energy, which is crucial for various biochemical processes in the living organisms, including electron transfer reactions and energy metabolism. So that's it for today. Thank you for watching. We are conducting a free quizzes on a daily basis in our Telegram channel. To improve your conceptual knowledge and ace your performance, do attend these quizzes and follow us for more videos. Thank you.